Hi everybody, I'm Justin Jarrell. I'm the VP of Business Development at TokuBio. I'm here at SITC in Houston, Texas, and I'm going to be presenting today one of our posters from Tako. This one is specifically on our validation of a whole blood collection kit, which we call the TokuKit. The TokuKit is a two-step, 20-minute kit for fixing blood that's suitable for cytometry analysis. Specifically, I'm going to be talking about mass cytometry, but keep in mind we also have this working for spectral flow cytometry too. So one of the biggest challenges with clinical trials today that are using cytometry in their PD studies is that these samples need to be processed live. And what does that mean? They need to be processed within 24 to 72 hours of the blood being drawn from the patient. And you can imagine that can be a pretty big logistical nightmare and challenge for these groups, is because such a short time frame for them to get that blood process. And so what happens is about 20%, if not more, of the samples fall out of spec. You can't use those data anymore, and therefore you're losing these potentially critical insights or biomarkers that are predictive of response and safety in those clinical trials. And so what we did is we created this kit in order to solve that problem. So now with our kit, which I'll describe sort of the process over here, groups are able to fix samples at the clinical sites and have those samples stable for 24 months in the -80. And during that period, the sample can be processed by cytometry. So you're going from 3 days to 24 months now. It's a much bigger window to handle for any clinical trial. So here's the overall process of how the TokuKit works. Again, it's 2 steps. It's super simple. You collect blood in either a heparin or an EDTA tube, about 2 mils of blood. You transfer that 2 mils into a buffer 1 that we call. That buffer 1, you close the tube, you invert it 10-15 times, and then you incubate it for 15 minutes at room temperature. After the 15 minutes, you then transfer that material from buffer 1 to buffer 2 with a provided pasture pipette, and then you put that either on dry ice to ship out to a central lab or to us at Tako, or you can put it in the -80 for long-term storage and send it over when the time's right. So in terms of how it works with mass cytometry, so we took that and did it on a 33 marker CyTOF panel, and we then wanted to see, hey, if you fix the sample, how similar does that look to a live blood sample? Because it's been known that if you do fixation on a sample, you could disrupt epitopes and prevent the binding of antibodies to those antigens. And so that was the first thing that we wanted to see. We took 3 healthy donors, live stained with the TokuKit, and we looked qualitatively at the dot plots, and we saw when we look at T cells, B cells, NK cells, monocytes, dendritic cells, and all their subpopulations. Generally speaking, when we look at the gates, they look very, very similar to one another. And then qualitatively, when we actually looked at the immune cell frequencies of the immune cell populations and the functional marker subsets, and we looked at the live sample versus the fixed sample, we see that they're highly correlated with one another with R values greater than 0.9. We also show that if the blood is processed, meaning used with this kit and fixed within 5 hours, that looks very similar to a sample that we process within 3 hours. So we recommend processing the blood sample with our kits at the clinical sites within 5 hours. And then we also show once that sample has been fixed and frozen, we show that 7 days after freezing and up to 24 months after freezing, when we perform the cytometry analysis on it, the immune cell frequencies between 7 days and 24 month samples are very similar, again with a high, I think it's about a 0.999 R value to that. And then what we go on to show is that when you take the kit and the CyTOF panel as an assay, and you perform precision studies on that, specifically intra-run, inter-run, and inter-kit tests, you get low single-digit coefficients of variations on those values, indicating that within this replicates, they're really reproducible, the results. And then we also do an inter-instrument kit where we compared it between 2 instruments, and we showed that when we're analyzing the frequencies of these immune cell subsets again, the percent change from one instrument to the other is very, very low at about 1.3% on average. So ultimately, we are presenting this because this is, again, a potential option to ease your workflows and make sure that you have more samples in spec for your clinical trials for cytometry analysis. Again, right now, we've got about 9 clinical trials that are currently using the TokuKits for their profiling, for their PD studies. And so if this is something that you're potentially interested in or you're facing these challenges too, please visit our website at tako.bio, or you can email me also at justin@tako.bio. I'd be happy to give more details to this and introduce you more to Tako. So with that, thanks so much.